Thank you. So hopefully this is all working. So welcome everybody. Thanks for coming. I figured for this talk I would talk to you guys just a little bit about some of the methods we're going to be using, because as we just said, data collection is still kind of ramping up for all of our projects, since we depend on the rural health cohort to get our data for my project. So we've done some pilot subjects, but we're still getting the users in there. So I'll tell you a little bit about some of our earlier work and kind of the methods that we use that like lead up to this stuff. Now this stuff is a little bit, I'm looking to see who's out here. It's a little on the technical side. It's, our stuff is very neuro-y, it's very computational. So I don't know how much that's everyone's cup of tea. Feel free to like slow me down if it gets too weird, or Siri really wants to talk to me about this too. Slow me down if something is too weird. I figured I'd kind of give a little bit of a skinny overview of the really technical stuff, but if you really do want to get into the equations and stuff and make everybody else miserable, let me know. Okay, so yeah, we're going to be talking about some of this multivariate pattern analysis and deep learning stuff, but before doing that, I figured I'd kind of give you a little bit of background on some of the classic work that I did kind of back in grad school and leading up to this stuff, using more conventional methods, and then you can kind of see where our interests started out. So one of the main focuses of my research and my lab's work is kind of the interface between what we call, well, perception, which, just vision and other senses and that kind of thing, and what we call reflection, which is not just kind of like the deep philosophical sense of reflection, but any kind of like internally directed mental activity. So that could be working memory, things like that, right? So one of the things we've done a lot of study of is something called, or what we call reflective attention, which is attention directed the way that you would direct like your visual attention, but how you direct your attention among your thoughts and memories and things like that, right? So it's pretty well established that attention can operate within reflection perception. We've had some work on that, and plenty of other people have established that too. And there's a particular operation we've done a lot of work studying, which we call refreshing, or just like a kind of one way to conceptualize this is an active reflective attention to a mental representation. So essentially that means like of all the things that you have kind of zooming in and out of your awareness inside your head at any given time, refreshing is what we call kind of like one little instance of you putting your mental spotlight of attention on one particular idea or concept or item of memory, right? Are we good so far with that? The basic kind of psychological definition there. Okay, so and this is thought to be just one component of much more like complex tasks and so on. And a lot of the work that I did in graduate school was looking at this one particular operation in isolation as kind of extracted out of a more complex kind of working memory task. And in particular, one of the questions that we were interested in, which we'll get to, will have some relevance for the work we're doing here with the RDAR Center, is in what ways attention, either perceptually or kind of directed internally in your thoughts, in what way attention can enhance or even suppress mental representations or like items in memory. So our one of the basic questions of the study I'm about to show you is, I'll just tell you that we know already that when you put your mental attention on something, it kind of enhances your processing of that information, right? So if I give you a face and a scene to remember, and I tell you to think of the scene, you'll have better memory for that scene later on. But the question is, what happens to the face that I didn't ask you to think about, right? And of course, our more fundamental question is like, what is attention, basically? And this is kind of a thing we think about a lot in the lab, like, is attention kind of a zero-sum game, like a seesaw, whereas if you kind of enhance one thing, you kind of push another thing down, or can you kind of boost something in memory without there being a cost to other information? That's kind of the question that we struggle with. All right, so I'll show you the design of this study. Again, if you're a cognitive neuroscientist person, this is like probably in your native language. If you're not a cognitive person, please let me know if I'm like forgetting any sort of like obvious things I should explain that we tend to be lied over in cognitive science talks. But this is the basic structure of a task we used for an fMRI study we did a while back now. And we had kind of three main conditions. The first of which we called refresh. So basically, well, somebody's in the scanner in the refresh condition. What would happen is they get a trial. It's about 5 seconds long total. And that trial would begin. I would use my pointer if I had a pointer here at this point. Nobody's got no Teddy Roosevelt's in the crowd with a big stick. So we would start with just a fixation cross. Just put your eyes here and don't do anything. Then for 1.5 seconds, people would see two items, which would be a face and a scene. So imagine you saw that kind of whatever that office or churchy looking building is in that woman's face. There's a brief gap of just 0.5 second, and then you would see an arrow pointing to either the left or right side of the screen. And what the arrow indicates in the refresh condition is that you should kind of briefly think back to, just put the focus of your mental attention on whichever item that arrow is pointing to the former location of. So in this case, the arrow points to the right. The thing that had appeared on the right was that lady's face. So you just briefly visualize for only about a second and a half that woman's face, right? Then it goes up the screen, trial's over. No responses or anything. This is just a purely mental activity. This is just saying, oh, just for a moment, put your mental focus on either the face or the scene. And we wanted to contrast that with what we call the attend condition, or like the perceptual attention condition, where it was basically the same stimuli, but you see the time order is now reversed. So here you get now a pre-cue arrow pointing either left or right. Then you see the face in the scene, then there's a gap, and then there's a fixation cross at the end where you don't have to do anything. So it's a similar idea, but now you're getting the arrow ahead of time to tell you when things come on screen to look at, only look at one of them and just ignore the other one, right? So it's still a form of attention, but now you're doing it prospectively instead of retrospectively. Okay, everybody on board so far still? And then we had kind of a third neutral control condition, which we called act, because what happened here is you would get the fixation cross in the beginning, the face in the scene, the gap, just like in the refresh condition, but then the thing that you got at the end would be this little gray square, and that little gray square just meant press a button. Don't think of either item in particular. This is just to give them a little something to do here so their minds don't wander, any more than necessary. And in theory, they're not thinking of either the face or the scene, especially in this condition, right? So this is the closest we can get to kind of a totally neutral perception of the face and the scene without special attention to either one. Okay. Everybody good so far? Any questions about the basic setup here? All right. So I said we have 3 conditions, but really, we're really going to end up having like 5 conditions, because each of those refresh and attend conditions can kind of break down in 2 ways, right? You can either be told to think of the face, or you can be told to think of the scene, right? And what we wanted to do is we wanted to look at activity in a visual brain area. In this case, I'm just going to show you area from the parahippocampal place area, or PPA, which is an area that's been studied extensively for the last 22 years or so. As an area that is more or less specifically active when people look at or think of scenes, and not when they think of things like faces. So what we're going to do here is we're going to look at brain activity from fMRI in the PPA in each of these different conditions, and use that basically as an index of how much the scene in a given trial is activated in those different conditions, okay? This is a pretty common thing to do in like working memory studies and so on, is use these, now that we know, we didn't take it for granted before about 2005 or something like that, that visual areas participated in working memory, but now that we know that, we can kind of use activity in these category-specific visual areas as like a proxy for what people are mentally attending to or fixing on. Okay, so what I'm going to plot here for you is activity in the PPA. The units are arbitrary units, so you don't need to worry about what they mean exactly, except that more of them means more brain activity in that area. And if we look at how much activity there is in that sort of baseline act condition, I'll kind of go in reverse order from what I introduced these as. There's a certain amount of activity in this area when you have this baseline condition. And that makes sense, right? Because in every condition, people see a face in a scene, so the scene area is going to have some non-zero amount of activity in every condition, right? And if we look at the condition where we just have you attend to the scene and ignore the face, just perceptually, not in your internal memory, you see that you get a boost in activity, right? And again, this makes sense because in the attend scene condition, you don't have to focus any attention on the face. You can just pay all your attention to the scene, and you get a little bit more activity in the scene area. As you might expect, if I tell you to attend only to the face, it drops relative to the baseline act condition. That's pretty much what you'd expect, right? Like if you've got a face and a scene on the screen, if I tell you to look only at the scene, activity goes up. If I tell you to look only at the face, activity goes down. The more interesting question is what happens when I have you mentally attend to this stuff after they've gone off screen. And what happens if I tell you to think back to the scene is you get an increase in activity relative to the baseline, very similar to what you see in the attend scene condition. So you get almost exactly the same amount of benefit to your sort of, well, what we think is the mental representation, but you certainly get as much benefit to the brain activity of that representation as you do if you're visually looking at it. And the real question is what happens if I tell you to think back to only the face, right? So there's basically 2 hypotheses here, one of which is that it doesn't change from baseline because it doesn't cost you anything to think of the face in terms of how the scene is represented. The other option is that activity drops relative to baseline because even thinking about the face will kind of suppress your brain's representation of the scene after it's gone on screen. So I'll give you a minute to like place your bets and decide which it's going to be. And the answer is you do see a suppression, not quite as much as if you're only visually attending to the face, but a pretty significant decrease in activation when you're thinking about the scene and mentally ignoring the face, right? So this is interesting to us, again, depending on how much you want to get into the weeds of this stuff. For the cognitive non-cognitive people, you may not be that interested in this stuff, but we're interested in this because it shows us that like, at least we think that this means like mental attention is kind of this zero-sum game where like it not only helps, it not only helps your representation of the face to think of the face, it actually hurts your processing of the scene, right? So this suggests that there's some kind of suppression or inhibition going on rather than, just kind of like leaving everything processing as if you hadn't thought about either item at all. Okay. So where it starts to get a little bit more interesting for what we hope to do in this project is we'd also run the same task in a group of healthy older adults. And so this is from a paper, again, a few years ago in the lab I did my PhD in. And again, looking at the PPA, what I'm going to show you here is we're just going to go ahead and subtract out the baseline. So now like 0 is basically the point that the baseline condition was, and everything else will be expressed as higher or lower than that. And so this is that same PPA area. And what I'm going to show you is kind of a little, on the left here, I'm going to show you a little kind of chart of whether the young or older group showed enhancement or suppression for these different conditions. And so if we plot these out, when we tell them to attend to the scene, you've already seen the young adult data, so that part shouldn't be a surprise, right? When we tell people to attend visually to a scene, you get a boost in activity. In both the young and older adults, they look almost identical to each other. So check mark for enhancement of activity when you think of the scene. Oh, sorry, when you visually pay attention to the scene. Stop me if I trip up on any of these because it's very easy to like misspeak. We do get, when we tell you to attend only to the face, we do get a suppression relative to the baseline condition, again, in both age groups. So again, young and old pretty much look exactly alike there, more or less. If we tell people to refresh the scene, again, we see significant enhancement above baseline in both the young and older adults in this area. Maybe a little bit less in older, but not enough that that's reliable, but they are both reliably above the 0 point. So again, so far so good. Young and old adults look exactly alike. But when we get to that condition where we saw that, suppression of the scene when people were thinking of the face, here's where the young and older adults look very different from each other, right? So the young adult data you already saw, when you think of a face, your representation of the scene is suppressed, but in older adults, it looks like exactly baseline, right? So they seem to show impaired suppression of competing information when they're told to direct their memory, to something else, right? And this is consistent with a lot of the kind of theorizing about aging, which, shows a lot of like inhibitory deficits and so on in older adults. And one of the big ideas there is that an inability to kind of like gate out information that you're not supposed to be attending to is one of the reasons that we see certain deficits in various cognitive tasks in older people. So that's interesting. And the reason that's interesting for us for this project is this is a case where we're taking a very simple mental process, right? Like 1.5 second mental activity, right? Which is a lot simpler than a lot of the memory tasks and so on people use. And we can get pretty well drilled down into a very specific deficit that older people have in this task. And these are, by the way, Alzheimer's or anything like that, just regular, active older people. It's a very specific deficit, and it's suggested that this might be a part of like why you see other deficits in more complex tests and stuff, right? But you really kind of drill down and get something very specific here. And of course, because one of the things that drug users are known for is not the greatest inhibition in the world, right? This is one of the many tasks that we think we might be able to see some inhibitory deficits in our rural drug user population that may or may not have been apparent in certain other paradigms, right? Because when you kind of drill down on these things, you can sometimes pull out things that you don't see in more conventional tasks and so on. Okay, so a quick summary of what we learned here today so far. So we learned that both mental and perceptual attention can enhance and suppress activity in these visual brain regions. Suggests that there is this kind of like natural competition among these representations, that when you help one, you hurt another. But they can be kind of distinguished, the mental versus the perceptual attention, that that in some cases one breaks down where the other doesn't. But anyway, so what we also saw though, right, is that mental attention or reflective attention might be specifically compromised in some populations, more so than perceptual attention, right? So, a lot of the literature on both drug use and aging suggests that like perceptual attention is actually relatively well preserved in both cases. You don't see a lot of deficits in traditional attention tasks in either older adults or drug users, but this particular mental attention operation might be more specifically compromised. And then they might be pretty subtle, but you really have to kind of drill down into these very fundamental cognitive operations to see these deficits appear. Okay, questions so far? I know this is kind of whirlwindy. Everybody hanging with me so far so good? To my lab and everything, this is old hat. But in non-neuro people, I don't want to blow anyone's minds yet because it's going to get hairy. Okay. So let's look at a second study. This is, again, segueing more into one of the other projects that we're going to, or tasks that we're going to collect in our class project, part of the RDAR Center initiative. So we've also done the same kind of thing in EEG, not just fMRI. So I'm going to show you an EEG study that we did again a few years ago that we're basically doing a kind of revised replication of. If people aren't familiar with EEG, that's the thing where you put the cap on your head. This is our then undergraduate. Now he's probably all grown up now and got a PhD and everything. I'm not sure where he is these days. But that's one of the guys that co-ran the task or the study with me. With EEG caps, so we record, electrical activity from your scalp at anywhere from 30 to 60 to hundreds of locations. In our lab, we were doing just about 30 for this project. And we get as opposed to fMRI, where you get a very slow measure of brain activity in a very specific spatial location, with EEG, you get a very spatially imprecise, but temporally precise idea of what's going on in the brain. So this was at the time, I believe, the first EEG study of this simple refresh process. And one of the basic questions we had was just, can we recover information about what is being refreshed using EEG? One thing we just didn't know was what it looks like in EEG when you do the simple refresh operation. We were kind of interested in whether that could give us any new insights as to how this process works. So I'll give you the basic task here and then show you a little bit of data. So this is fairly similar in structure to the task that I just talked to you about from the fMRI study. A little bit changed up here. So in this particular version, what we did is people still saw 2 items on every trial, but this time there were always 2 items of the same category. So people would see either 2 faces, 2 words, or 2 scenes, again, for 1500 milliseconds. So you just kind of look at those and encode them into your memory. There'd be a 500 millisecond gap where nothing would happen. And then for 1500 milliseconds at the end of the trial, you get one of 2 cues. There's actually a third kind of cue that we're not going to talk about here. But you get one of 2 cues, one of which was basically just like a blank or a green dot that we called no act, because basically you didn't have to do anything there. They just kind of kept looking at this. Or you get an arrow, kind of similar to the other study, where what you would do is mentally think back to one of the 2 items. So there was no perceptual attention condition in this study, just a refresh or nothing, basically just refresh or a baseline condition. And so what we're going to be looking at here in all the data that I show you, I mean, we have EEG activity for the whole task, obviously, but we're only going to be looking at the events here where the cue period is. So that 1500 milliseconds where either an arrow or a dot is on screen, right? So I'm going to show you brain activity, again, just in one electrode. We've got 30 electrodes, but I'll just show you the one that's kind of the most interesting, or where we expected to see the most interesting stuff going on. And this is what happens. So to just orient you a little bit, if you're not used to looking at EEG data, this is a head. It doesn't look that much like a head. A head is just, in EEG parlance, I guess, a circle with a triangle on the top representing the nose. So these are the names of all the electrodes that we recorded from. The one that I'm showing you here is called T6, which is kind of right back here. And if there's any serious EEG people in the crowd, don't shoot me for saying this, but you can think of this as roughly analogous to some of these areas like PPA or FFA that respond to faces and scenes. So it's the activity we see here, we think kind of reflects activity in some of these higher level visual areas to a first approximation. And so we were kind of interested in seeing what would happen in an area like this when people were thinking of things like faces and scenes and words, okay? So activity from this electrode back here on the right side of the head. What we're seeing here on the left is a plot of voltage over time. So this is the voltage in microvolts. So it's a very tiny signal, but when you average enough of them, you can get a stable response. And what we're seeing is the 1500 milliseconds from when the cue comes on screen to when it goes off screen, plus a few hundred milliseconds at the end, after the cue goes off screen. And what you see here is the green plot there shows us what's going on in this area when they get the neutral green dot Q, which is, you get like an initial dip down into negative, then a positive peak, and then it kind of dips down again and so on. And one of the big questions of the study was just, well, does it look like anything when you mentally refresh stuff? And the answer is it does, right? So you see the differences appear pretty early on. That very initial difference before about 200 milliseconds is probably mostly from the visual difference between the dot and the arrow. But then we see also later potentials that are more cognitively associated, which at about 400 milliseconds, I would get it right up on there, but I'm kind of boxed in by cables here. So you see a higher initial peak at about 400 milliseconds. And then what you see also is this weird kind of like sustained activity between about 800 and about 1400 milliseconds, where there's sort of a more positive EEG response in the refresh conditions than the baseline no action conditions, right? And by the way, the bold time points here, if you can see the difference between the bold and the non-bold on the screen, the bold ones represent the places where this is significant at a corrected threshold. So much of this timeline is like individually significant at an FDR corrected statistical threshold. So what this suggests to us is that there are at least two ways in which this mental operation of refreshing differs from like a neutral non-refreshing operation. One at this initial peak of about 400 milliseconds and one at like between 800 and 1400 milliseconds with a smaller but more sustained response. And we have some theoretical ideas about what that means. I mean, our thinking is that like that initial peak relates to the kind of initiation of like, oh, what am I supposed to do? Oh, I guess I should refresh. The arrow means this, okay. And what we think that more extended what we think that more extended later activity refers to is probably similar to what we see in posterior brain areas, like in the fMRI study, where we think that probably represents the modulation of these like visual areas by actually kind of reactivating that memory. So there's various theoretical reasons we think that, but that was our interpretation of this so far, right? Okay, so if you're not really into the weeds on this stuff, the main thing I think to take away from this is that, there is something going on that we can detect when people do this very short mental operation. And keep in mind, this is only for just refreshing in general across all the stimulus categories. I haven't shown you yet what happens differentially when you refresh one category versus another, right? Does this basic plot, this basic data make sense to everybody? Are we on board with this so far? Again, I know for people in like my lab, you've seen this stuff too many times, but for everybody else, I don't want to like get too crazy too soon. Okay, so here now is the same kind of thing. So I took away the baseline condition. I'm showing you only the refresh conditions, but here are the refresh conditions broken down into what category people were actually refreshing, right? Because they could have been refreshing either a face, a scene, or a word. Here are the plots. You will notice that they look very similar to each other, much more similar than the refreshing and baseline conditions look to each other. So in a sense, that is good, right? Because it suggests that this is kind of maybe a similar mental operation across different classes of stimuli. But in a sense, it's bad if we actually wanted to be able to like know from brain activity, if we want to like do some mind reading or brain reading and know from the brain activity which of these items people are actually thinking about, right? So we can all stride further into our dystopian future where everybody can know what everybody's thinking all the time. Facebook would love this. So you see there are some bolded time points here, but in this particular plot, the statistical threshold is much, much lower. So those bold time points here only represent uncorrected p less than 0.05 time points. So nothing in this analysis differentiated between the different stimuli or the different categories of stimuli at a corrected threshold. So basically, if we were being statistically rigorous, there is no difference between refreshing these three different conditions, right? Which again, like, kind of good for the robustness of this process, kind of bad if we wanted to actually know something about the category from the EEG signal. Okay, and again, this is pretty characteristic of what we see at lots of electrodes. I'm just showing you this one rather than all 30 of them at once. Okay, questions on that so far? Everybody still on board? So we did not, well, actually, yeah, so we did not give up yet about the category information. So, so what we wondered is, can we use this somewhat more sophisticated, more recent type of analysis called multivariate pattern analysis to recover that category information? So is there any way that we can kind of squeeze a little bit more out of the data and figure out what category people were refreshing based on the EEG activity we have? And to talk about what we found, I'm going to take a little detour into explaining what this MVPA stuff actually is. So just to give you the conceptual overview, so if I can kind of like simplify the classical neuroimaging approach a little bit, And it's probably best, I don't want to like give you mental whiplash here, but it's probably best if you think about this in like fMRI terms, where you get kind of like a picture of the brain and like blobs of activity and stuff. I think people have probably seen more of that than they've seen EEG data if they're not EEG people. So the classic neuroimaging approach, what you see in journals is you see these areas like the fusiform face area, which is abbreviated FFA. That's an area like the PPA, but this one likes faces and doesn't like scenes. You've got other areas that respond to things like objects. So the way that people probably mostly think about fMRI and neuroimaging is, okay, you do some brain scans and you find these little like blobs of areas that respond to like faces more so than objects or whatever, right? People have probably seen plots that look something like this in a journal, right? Where you get these little blobs of activity, right? And so a lot of the early neuroimaging work was like focused on sort of blobology of going like, okay, I found a little chunk of brain that likes faces, and a little chunk that likes objects, a little chunk that likes bodies, and a little chunk, a lot of the visual stuff focused on this kind of thing for a while there, right? But the MBPA approach is to say, well, let's dig a little bit deeper, right? And say, like, I don't just care about sort of this overall area that seems to respond to this broad category. Let's try to zoom in a little bit and see if like, maybe there's not a whole area of the brain that responds to just chairs or just shoes or whatever, but maybe, we can see from the specific pattern of activity in one of these areas, we can decode whether somebody's looking at, say, a chair versus a shoe. So if you can imagine that we took this purple area from the top of the slide, this object area, and we zoomed in on and said, okay, well, if you kind of look specifically at what's going on in this purple area at different trials of some visual task, imagine this purple area is really composed out of a bunch of, in fMRI we call them voxels, which are like pixels, but 3D volume elements instead of pixel elements, so voxels. So you've got, this purple area is really composed of a bunch of voxels that are like either blue or red in this kind of, metaphorical depiction, where, red, blue make purple, right? So like the idea is that like, if you kind of zoom in on this pattern, you see that what looks purple at a broad perspective is really made up of a lot of interdigitated blue or red, and that what you might have is like individual single sort of tiny little bits of tissue within this broadly object-sensitive area that respond more to chairs than shoes or more to shoes than chairs, right? So the whole area doesn't seem to care about whether you're looking at a chair versus a shoe. But if you zoom on in, individual voxels might care a little bit about whether it's a chair or a shoe, right? And the signal in any one voxel is not very strong or stable, but if you can aggregate across them, you can kind of make this overall prediction based on the pattern of brain activity you see, whether something's a chair or a shoe, right? So the idea here is like, well, if I kind of collapse across a bunch of different trials, I have a little animation here of like, imagine you were looking at a bunch of different trials. With enough trials of activity, you can kind of see, okay, certain voxels semi-stably, like when they're active, it seems to indicate that there's a chair there. Other voxels, when they're active, it seems to indicate that there's a shoe there, right? So the idea is basically to say, for a given, sample of brain activity, we add up kind of how many of the red voxels are active versus how many of the blue voxels are active, and whichever one is sort of, act more active or sort of voting harder for that category, we guess that that's the category people are perceiving, right? Does that kind of make sense? So this is what, when people talk about decoding brain activity, this is kind of what we mean here, right? Is that you're sort of aggregating a bunch of tiny responses to make an overall prediction rather than just looking for a whole blob of activity that all just means one thing, right? Okay, any questions about that before we go on? All right, so now, and if that doesn't make sense to you, just, it's magic, okay? So that's also, it's also a valid mental model for what we're doing, is magic happens. So we put our activity or our data into our like magic computer box, and so what we saw previously, right, is we saw like these plots of EEG activity where like refreshing a face versus a scene versus a word, look very similar, but can the magic box, do that better than just looking at those plots of activity? And so I'll show you what happened when we did this, and essentially what happened is. And don't worry about the measure too much again if you're not too mathy, but this is area under an ROC curve. So basically anything above 0.5 would be above chance. And what we see basically is when we look at the full epoch of data, and this is aggregating across all the electrodes, every part of that 1500 milliseconds that people are doing the refresh task, we do see that we're a little bit above chance of predicting what category people are mentally thinking of, right? Which is, I mean, again, I don't know if you're impressed by this, if you don't do this stuff, but we were impressed by this because this is decoding, purely from internal thought, like what people are thinking of for only 1.5 seconds, in a pretty subtle distinction. So that was pretty cutting edge at the time. So good news, everybody's really happy. Then we had the thought like, well, we should probably look at that neutral control condition and kind of see what's going on in there. And unfortunately, if you can think back to like how the design was set up, we actually can also decode above chance from that period when it's just a little green dot on screen. We can decode which category people had previously seen in a trial, even when that green dot was on screen, which was a little concerning, because that means like, oh no, well, maybe we're just decoding like some kind of residual visual activity from when they first saw the faces and scenes and words. Maybe we're not actually decoding what they're thinking of. So we got sad again. But then, and this was actually my advisor's idea, so full credit to her for this. She said, well, you know, let's split out. We saw some like early components and some late components to this EEG signal. Let's split the early from the late and see if that makes any difference. So if we look at only the first 800 milliseconds of the trial, we see basically the same thing. In the first 800 milliseconds of any given trial, in both the refresh condition and the baseline condition, you can decode what category people had previously seen. But if you look at the last, you know, 6 or so 100 milliseconds of the trial, we see a glorious separation here, right, where the neutral or baseline condition goes totally to chance and refresh stays high, right? So that leads a lot, or lends a lot of support to our hypothesis that that later period represents kind of the modulation of these working memory representations, and that you kind of really need to be actively working to sustain those. And if you don't, that just kind of goes away, right? So we think the first half of the trial, yeah, may have been like lingering visual activity, but the last half is really what's going on when you're mentally kind of sustaining or reviving those representations. So that's great. So we can decode what we think is a true working memory representation in that last part of the trial. This is just a timeline kind of the same thing. It's a little messier because what we're trying to do is we're trying to do the same classification analysis on very small chunks of data from discrete time points. So this is a time point of classification using only little bits from a particular point in time. But what you can see is, roughly speaking, like right here around 700 milliseconds, there's a real distinction between the refresh condition and the baseline condition, where refresh actually goes up and the other one goes down in terms of how well you're able to classify what people are thinking of based on activity at that point in time. So it's a lot messier, but I think it still makes the point. Okay. So in summary for that study, if you're keeping up here, so we found that these, like, you know, we've already split like more complex working memory tasks into these very simple, like 1.5 second long mental operations. And this EEG study shows us that we can further split it up into at least 2 temporal components, kind of an early and a late component. And we think, you know, for various reasons, like I kind of alluded to earlier, we think the first one likely corresponds to more frontal activity when you're trying to decide what to refresh and, you know, where to direct your mental attention. And the second to this more posterior, the actual representation of the item in working memory. And we can recover category information during this process using NVPA, and it seems to be refresh specific during the later parts of the trial. So that's pretty neat. Again, a nice case of like showing you kind of like when you really drill down, you can get a lot of information out of surprisingly like small pieces of data, right? All right, so that's most of that study. Questions about that? Is that making sense to people more or less so far? So this is another one. We've never done an EEG study before in like aging or anything like that in a special population, but we expect this stuff to show kind of the same sorts of deficits we see during fMRI. So this is another study that we're going to do a kind of revised version of for our radar center activities. Okay. All right. So if you're with me, is everybody awake still so far? Still good? All right, because I haven't gotten to the hard stuff yet. So let me tell you about a little bit with the time that we have left, a little bit of the kind of more technical, more exciting stuff that we've been getting into recently since I got to UNL. And a lot of what we've been doing has been using this deep learning stuff. People have probably heard about some of this in news and things like that and papers and so on. This is all the stuff that you know, all these companies like Facebook and so on are using to, you know, predict everything you're doing. If people are familiar a little bit with artificial neural networks, deep learning is basically just kind of a revival of that work with more complicated neural networks or fancier. I won't go into the details, but the basic idea here is that you simulate in a very rough sense computationally kind of what a neuron does, right? So what a neuron fundamentally does is it aggregates a bunch of inputs, you know, that come in the form of dendrites. It sums them up in some way, and if those inputs from those dendrites are sufficient to produce this neuron or to cause this neuron to fire, it fires, right? And you get this output that is either a yes or no, like the neuron fires or it doesn't, and of course that feeds into other neurons and so on, right? So the earliest artificial neural networks were really not even networks, they were just artificial single neurons where you put in a bunch of input values, you sum them up as multiplied by a certain weight, and if the sum of all those weighted inputs exceeds a certain activation value, you get like a 1 versus a 0. And if you are a mathy type person, you may notice it looks very much like, this looks very much like a regression or like a logistic regression, right? Where you figure out what the betas are, the weights associated with each of your input values. You know, for a given trial or observation, you sum them up, and, you know, if they cross a threshold, you say yes, and otherwise you say no, right? So that's like a single artificial neuron, kind of what it does. And, you know, you can kind of combine these into more complex networks. So what you might have is something like, you could have like 4 input values and then a bunch of neurons kind of interconnected with each other. Those are the little blue dots. And then multiple different possible output scenarios, which are the little red dots or the Ys, where you would say like, okay, you know, I have a bunch of different input values, and then the numbers get crunched by these artificial neurons in lots of ways. And then depending on which of Y1, Y2, or Y3 is more active, we'll make one of 3 different predictions as to like what that input pattern represented. So you can get all kinds of variations on these things. And then there's like a zillion different flavors of neural networks that people have come up with, especially in the last decade or so, that I will not trouble you with. But the basic idea is you can kind of like make a little, tiny little mini brain that is super simplified from a real biological brain, but it's, you know, you can train it to like make certain kinds of decisions or do certain types of computations based on pretty much any sort of input data you're willing to give it, right? And so this has been used in lots of stuff that you may or may not have seen before. So a lot of this is what's being used now for like image tagging. So if you do like a Google image search now or something like that and search for, say, a jack-o'-lantern, you know, this is, you know, they've trained these algorithms to like recognize and tag images. And these get as complicated as things like these are computationally derived descriptions, like black and white dog jumps over bar, like that, you know, a computer came up with that, right? So it works pretty well sometimes. I don't have any of the like hilarious fails. It fails hilariously sometimes too, but it's what enables a lot of the stuff. Like if you've ever used one of these translation apps where you just point your camera at something and it translates it, all the Google Translate stuff like that is using these deep networks too. You can do things like artificially colorize, you know, black and white images. People have probably seen like all the stuff about deep fakes now, right? Where you can basically put anyone's face on anyone else's body video and, you know, come up with some really disturbing, you know, fake news or whatever. So you can do all kinds of this stuff, right? And people have gotten really excited in the psych field also because some of the these artificial neurons that you get when you train out these networks sort of look like the response patterns that you get from real neurons sometimes. And so people got excited about that, but that's actually not what we're doing here. What we're mainly interested in deep learning for is using it for data analysis, not so much as like a comparison to how human beings process data, but just to see if that can like get more out of our data than we can get otherwise. So I'll just kind of, I don't want to take too much time on this, so I'll just kind of go quickly through some of these pros and cons here. Some of them you will see are kind of the same. So one of the cons of deep learning is that it's more complex, so you can get more sophisticated analyses. So, sorry, I got this backwards. That's one of the pros. The con is it's more complex, so there's lots of different like things you can try. You can kind of like get yourself into the weeds, trying a zillion different kinds of neural networks to see if any of them are better than the analysis methods you're used to. So, you know, that's a pro and a con. This is a little bit paradoxical, but one con is that it's slower. So these are much more complex analyses. So like per trial, they can take a lot longer to like train these up than like a more traditional analysis method. But the pro is people have spent a lot of time and money and effort getting these things optimized to run on things like GPUs, which can accelerate the process a lot. And in certain contexts, these algorithms actually converge faster than traditional MVPA. So they can be faster, even though computationally they are much more complicated. In practical terms, they can end up being faster to run. One con that doesn't really have a pro is that it does require you to have specialized hardware, not super specialized, but if you have the kind of video card you could play some really sweet games on, then that's good for deep learning. Definitely not using them to play games. And you know, and one pro is that you can just do some stuff with deep learning that traditional MVPA can't. There's just certain like shapes of data and types of analyses that you just can't do with traditional analyses, but you can with deep learning. And this next one is kind of a pro and a con together again, which is that because it is a more complex kind of analysis, you need a lot more data to give it in order for it to kind of get reliable results. On the other hand, if you have a really giant data set, that tends to choke some of these traditional analysis methods, and these deep learning methods actually become more practical. So, you know, for things like neuroimaging, where you can have like, you know, even a single subject, gigabytes and gigabytes of data, you can actually do a lot with these deep learning techniques. Okay. Questions or thoughts, or anybody curious about general deep net stuff before I talk a little bit about our stuff? Okay. How late do we want? Probably not beyond like 5 or 10 more minutes, right? Something like that. Or, yeah. I don't want to take the whole, if you're really interested, we can talk more about this, because yeah, I can kind of skim through this. So we spent a lot of time basically building a toolbox to do this for. So there's all these other tools out there to do deep learning, but to do it for like neuroimaging data, we've kind of built our own. I should mention that Carl, who's here, and Jake Williams, who is not, have been some of the main coders of this toolbox. So we got a nice little, actually Carl's working on a GUI right now. Mostly we just use kind of text files to program it up, and it lets us basically, you know, in a much more faster and sophisticated way, like helps us wipe out faster and better than we could have before. So we've come with this whole toolbox, you know, and now we got to figure out something to do with it. I'll just kind of show you a few of the main projects from our lab that have involved this stuff. So one of them is actually not using neuroimaging data. One of the nice things about this deep learning stuff is you can apply it to like any kind of large data set. So we have a pretty big data set of eye movements that we got from Mike Dodd in the psych department, and Zach, who is here, Zach Cole has been like applying this toolbox to some of these things. So one of the classic questions in like the eye tracking literature is, can you decode what task someone is doing from their pattern of eye movements, right? So we show you a bunch of scenes, and in this set of experiments, the things that they could be doing is they could be doing like a visual search versus trying to memorize the images versus like rating them for like how much they like them, I guess. And you get these like, you know, if you do some eye tracking, you get these like eye tracking traces that we've represented as like images here in black and white of like, okay, the eyes went here and then here and then here, and so on, right? And so the question is like, can we tell from this eye movement pattern like what task someone was doing independent of like what actual picture they were looking at? And sort of the best models that are informed by like all this cognitive theory and stuff tend to do about 50% or so in a sort of 3-category task like this. So chance would be 33%. And it turns out we've done it with a couple different formats of the data. One using like an image of the eye tracking data that looks sort of like that. One using just kind of like the raw eye tracking data, which is called timeline in this graph. And it turns out that actually it works a little bit better to use the raw data than the process data, but we get the same kind of accuracies that you get from these like much more sort of like theoretically informed models just by kind of throwing this into a deep learning model and saying, figure out what you can from this. So we're getting up, you know, our better classifications are in the range of like 50 to 54% accurate, which is pretty good, it seems like, for this field. And that's pretty neat because we don't need any theory, we don't need any sort of special effort to get that. And we can derive some things from this. So again, I won't go into super details, but we can like sort of then start to do things like pull apart, like, well, if you look at only the X data and not the Y data, or if we look at, you know, take out the pupil size data from the eye tracking and so on, we kind of start to look at like what things inform that more than others. So that's one of our current projects. One we call, it's hard to get a good name for stuff, right? A good like acronym. So we call this classification as bad epic rejection, or CABR. And the basic idea here is that we're using this as a way to clean up our EEG data by using deep learning classification. So you know, CABR is the acronym, and you know, when you reject a trial for being bad, we call it CABR tossing, like CABR toss there. This is a little conceptually tricky. So this is work that Chang Lim in the lab has been doing. She's back there as well. The basic idea here is that if you have a factorial experiment design, and this is just kind of a theoretical example here, and you're interested in a question like decoding whether people are looking at like a gray or a white object, but there's a second dimension to the design, like whether they're circles or squares, you can basically train up a classifier to decode whether they're circles or squares and say, okay, well, any trial that I can't tell if it's a circle or a square, that's probably not a very good like set of data or a piece of data. So I'm gonna like just throw that trial away and not use it in the classification I really care about, which is like decoding gray versus white. So there's a whole, this is a very nice figure from her poster and the paper that we're working on that I'm probably not gonna go into for time reasons, but there's this whole process where you kind of use one classification as the decision criterion for whether to keep certain trials or not. And then you essentially like see how that affects the classification performance you really care about. So I will just, if you're interested in that, we'll talk to you more about it later. But if we look at that same data set, this is one reason I showed you this EG data set earlier, that same data set where people were refreshing faces, words, or scenes in EEG, and then refreshing them. We've kind of got a good design for this where I didn't really look at data before from the perceptual trials, but we have trials where people are perceiving faces, scenes, or words. And then we have trials where people are refreshing faces, scenes, or words, right? So we have this kind of factorial design that works well for this kind of analysis. And what we see here, and again, I don't want to get too into the weeds with how this works, but in certain versions or certain formulations of this algorithm, what you see here is if you remove up to, so this is how well we're able to classify Oh, I should have put this in here. Let's put the other graph in here. Oh, Chang, you're going to kill me. Let's see. This is the top one's perception. The bottom one's refreshing. Sorry, I forgot to label that here. So in perceived trials, if you kind of do this initial classification first, your baseline at being able to classify what category people are perceiving based on EEG activity is about 57%, something like that, that first data point there on the graph. But if you remove up to say like 50% of your trials with this Haber process, you can get that up to something in the neighborhood of like 67% or something like that, which is a pretty big boost. And especially for this refresh decoding, which is what I showed you earlier, you know, in the traditional techniques and in kind of our baseline for this analysis without removing any trials, we're getting like 35% accuracy decoding what item or what category people are thinking of, which is above chance, which is down here, but not by much, right? But if you use this Haber process to filter out up to 50% or more of your trials, it actually peaks at like, what, 70%, but that's getting a little dicey as to like whether you're removing too much data. But if you remove a bunch of trials with your Haber process, you see that that goes all the way up from like about 35% baseline up to 40% or more, right? Which is, you know, still not 100%, but it's way higher above the chance baseline. So we think this is a pretty cool technique for people to do this classification and prediction type stuff, that it really can get you like a lot more usable. It's a little bit, you know, counterintuitive, but by cutting out half of your data, you actually can get much more reliable classifications. Okay. And for, all right, so for time's sake, I'll just give you like a sort of quick rundown of this stuff. We also, this is like another totally new kind of analysis method we basically like been able to invent using this deep learning stuff. This is kind of the old school version of classification that I showed you earlier, where you basically like throw in a bunch of data and you try to make a category prediction. This is just like drawn from some classification paper where you say, okay, well, if I want to predict whether something's a pedestrian versus a car versus a truck or a motorcycle, you know, you have this neural network with 4 outputs, and it you know, has an output for every category, and whichever category is the most active, or whichever output neuron is the most active, you predict that category. Again, I don't need to get into math and stuff for this, but that's the basic idea. Our new idea, which is really kind of an old idea retreaded, is maybe we train a classifier not to classify what something is. Like I feed in one trial of data and it tells you whether it's a car or a truck or whatever, but instead I feed you 2 trials of data into the classifier, and it does its magic, and it just tells you whether they're the same thing or different things, right? So it doesn't know anything about categories, it just knows how to tell whether things are the same or different categories. And it seems maybe like kind of a fine distinction, but it's basically using what I call the Sesame Street principle. If anybody remembers, the like, one of these things is not like the other. So you basically train a classifier to just know whether things are same or different, right? So if I clip like the actual letters and numbers out of this, like you can see at a very basic visual level, like one of these things is not the same as the other, right? So you can still use these category or these classifiers that kind of are trained to just detect differences to do categorization by kind of comparing. If you've got a W, you can kind of compare it to a corpus of known 2s, and if it's too different from the others, you can kind of essentially use that as a decision that that is not a member of the 2 category. So we did a little bit of basic validation work on this, which I will just kind of skim over. Not that, well, Jake's not here, so whatever. He doesn't get my compliments, but this is from a paper we submitted with Jake Williams, a student in computer science who got us started on a bunch of this stuff. Did some basic validation work and showed that basically this process can get us similar or sometimes better levels of decoding, again, using this kind of EEG data set we've been looking at a lot as our kind of test data set. Shows similar confusion matrices to traditional methods, so it doesn't do anything too weird. So it is basically validated. But the really cool thing I want to show you, because I think, not that Jake's work isn't cool, but the next step, which is even cooler, I think, is we had this design that we ran a couple years ago, an fMRI design that we wanted to use this technique on. It's actually the reason that we kind of invented this technique, where people were watching movies in the scanner. And basically we wanted to see like how well we could decode like what, in a very complex thing like a movie, what we can decode out of people's brain activity while watching that movie, right? So people watch these 6-minute videos. One was the first episode of Pushing Daisies. One was the opening to the movie The Brothers Bloom. People saw this multiple times in the scanner. They were just kind of watching attentively. And critically, there was also some resting state data. So we had some runs where people were just resting quietly. And what we ultimately wanted to be able to do with this data set, or one thing we wanted to be able to do, is say, can I tell from like one trial, like 1 second of brain activity, what moment in this movie people are seeing? Which, I don't know if you think that's cool, but I think that's cool if the answer is yes. Spoiler alert, the answer is yes. So So I won't bother you with all the math stuff except to say one of the interesting things you see. So what we're essentially doing with this paired trial classification technique is it essentially works out to like being a similarity metric. So it says whether things are the same or different, but we can also pull out from it like how much they're same or different. So it's very similar to what you do with something like a Pearson correlation or Euclidean distance or whatever. But the key thing that we showed when we started looking at this with the video data is you can get so what we're seeing here, again, I wish I could sort of get over there, but like if you look at these similarity matrices, like the top row there on the left is when we're looking at like one run of a video to another run of a video, so people saw these videos multiple times. So what you should see, if there's good signal in the data, is like a hot diagonal, and then everything, like lower numbers and lower values on the off diagonals. And you see that in our PTC technique, but you also see it with like traditional similarity metrics like Pearson correlations and Euclidean distance. The interesting thing is that if you compare something like a video run to a rest run, which is that second row there, or two rest runs to each other, what you should see is basically nothing, right? Because a given time point in a video run shouldn't look like anything in a rest run because they're just resting. And two rest runs shouldn't look like each other because what you're thinking about through the course of the rest run shouldn't really be similar between two rest runs, right? But what we see is like the traditional similarity methods like Pearson correlation, Euclidean distance show a lot of like temporal structure to the data that's not there in our paired trial classification classifier. And we don't need to get into like what these exactly represent over here, but we see a similar thing here where there's like temporal structure to the data that Pearson correlations and Euclidean distance are picking up that they really shouldn't. It's not meaningful data. It's probably something along just picking up like how the scanner is sort of drifting over time or how people are getting restless as the longer scan wears on or whatever. And our method is not picking that stuff up. So I'm going to skip over this for time. But the basic thing we did with this is to say, all right, can we use sort of like the sort of highest similarity values in these similarity matrices to say, if we have a given time point of fMRI data, based on the time point in a different run of data, what is it most similar to? Like how far off are we predicting which 1 second of that 6-minute video we are? So these are the histograms of our prediction errors. And so it works pretty well. The modal prediction error is 0 seconds, right? So most often we are exactly right. And pretty often we're within like 5 or 10 seconds. And then there's a long tail. I think the average is something like 9 seconds error or something, which is pretty good given that there's like a long tail of like, people are just gonna be asleep some trials or whatever. We actually do pretty well with that with both our technique and the Pearson correlations and Euclidean distance. But the key thing is like based on that validation stuff I showed you earlier, we think we're getting the right answer in our technique for the right reasons. We're not just picking up on like the fact that it's later in the scan. We think we're actually picking up on real visual information or real sort of brain activity that codes what moment in time people are seeing in the video. So I think I'll probably like wrap up there actually. We've got a lot of other stuff. So the reason that's interesting, I think, is we're going to use that in the class project too, because basically this is just, it's a very rich data set that you can pull a lot of stuff out of, right? So there's a million things we can do with this besides just what I showed you here. So it's a really rich data set for pulling stuff out of different populations and so on. So just to sort of remind people what we're doing here. So we're going to be doing 2 days of data collection with one fMRI session, one EEG session each day, featuring some of those same paradigms we saw today, collecting a bunch of different outcome measures. And we are going to basically be using a bunch of these deep learning models to basically identify features that distinguish users from controls, and then basically use those features to predict outcomes, right? So we've done a lot, we've come up with a lot of like cool techniques, I think, and we are itching to now apply those to some of our rural and quasi-urban drug users. So I just want to quickly say thanks to the lab. I think hopefully if I forgot to shout somebody out when I talked about their work, sorry. I realize I need a less outdated lab picture because that is an old lab picture, but thanks to all of you guys. Thanks to all our collaborators on our other big, massive, annoying, complicated grant, which a lot of the groundwork was these guys. I didn't put up pictures from the center because I figured you were all here, but especially like Kirk and Dev for all their logistics work and everything, and James, of course, for being my primary mentor. So thank you all, and thank you all for coming.